In this video, we're going to take a look at how biologists go about determining what some of the evolutionary relationships among bacteria are. So let's hop right in. So first off, let's look at some of the characteristics of the DNA, because that's a huge piece of how we do try to do systematics work in bacteria. So once we thought that bacteria only had a single circular chromosome, and that was it, and that every time the cell divided, it passed on a copy of that circular chromosome by binary fission. But now we know that some bacteria have multiple chromosomes, and that the chromosomes aren't always circular. Sometimes they can be straight chains, so a length of DNA rather than a circular piece of DNA. And when they do possess multiple chromosomes, then it's rare that each chromosome has the exact same genes in it. So they're not diploid in that sense. They don't have two copies of every gene, but instead they have, their genome is spread out across multiple chromosomes. And then we have all these plasmids that can be found inside of the bacteria as well. Plasmids are just little hoops, little circular stretches of DNA. They're usually much smaller than chromosomes. And when a bacterium divides, not necessarily, it's not necessary that both of the cells produced are going to receive all of the whole complement of plasmids that are found in the parent cell. Even within a single species, some of the individuals may have a single chromosome, while others have all of the genetic DNA to run the cell split up between two chromosomes. And then, as we've talked about a little bit, there is genetic transfer among individuals of the same species. What we talk about is horizontal gene transfer. So if we think about binary fission, where one cell produces two daughter cells, and then these daughter cells produce daughter cells as well, this is what we think of as vertical transmission of genetic information. But if we have in a culture two cells, and they don't even have to be the same species, and they do some conjugation, and they share genetic material with each other, then that would be horizontal or lateral transfer of genes. So anytime we have two currently alive different individuals that transfer genetic material, that would be horizontal transfer. Whereas passing on to a daughter cells is vertical gene transfer. So how do we do this job this job that is now part of systematics, where we try to infer or to make strong statements about what we understand the evolutionary relationships among the bacteria to be? Well, for comparison, in eukaryotes, we can use body shapes and body structures. We can use the fossil record. We can use the genetics. All of these are useful tools. But when we start looking at bacteria, morphology provides almost no, there is some information. We can do like gram-positive, gram-negative. But to really get down to the finer dividing amongst groups, morphology doesn't provide much information. And because they're so small, fossil record is essentially non-existent. So there are some big exceptions to this. Stromatolites that we just talked about, the cyanobacteria, are one example of an exception. And when we can find trace amounts of organic molecules, then that tells us a little something about living organisms in that area as well. So what we really have to rely on are the genetics, the DNA and the RNA that are found in the bacterial cells. And then the kinds of metabolic processes that the cell does and that the cell engages in. So the chemical reactions that occur inside the cell. So there's two big categories then of the kind of genetic information that's available to us. One category is numerical taxonomy, and the other is looking at genetic homologies. So numerical taxonomy we're going to hit in this lecture. Then genetic homologies, we'll talk a little bit about some, we'll introduce the idea of genetic homologies, and then we'll really unpack that in the next lecture. And then last of all, there are some other techniques that are also useful. Come on, little pen. There we go. That are also useful for getting information. And this is where we can look at some of the metabolic characteristics and properties of the cell. So numerical taxonomy is the first idea. And as we can gather more and more genetically determined information about the organisms that we're trying to classify, then the more information we have to work with, the more little data points we have around each individual, then the greater accuracy, or what we assume from numerical taxonomy, is that the greater the accuracy of the inferred, or the genetic relationship that we're trying to understand. And so in building cladograms, and this is an example of a cladogram here, where at the end of each one of these, so G, F, H, I, A, B, C, D, those organisms are all, those are the organisms. And then here are genetic traits. I don't understand why I'm getting the little, I think I know why. So all of those are genetic traits. And as we can gather information, does this individual have trait 14? Do any of these other individuals have trait 14? As we capture that information, then that information can be used to build evolutionary trees called cladograms. And so you look for the organisms that have the most shared characteristics and assume that they are the most closely related. And then using computers, we can build hundreds or even thousands of trees, these cladograms, and then we can classify them and apply some statistics to them, hence the idea of numerical taxonomy. And the statistically simplest tree is then assumed or judged to most accurately reflect the actual evolutionary history. And so every time we collect a new set of data about all of the organisms, then we can rebuild this tree. And each time we add more information, the idea is that we're getting a more accurate tree. And so this is a big piece of one of the, of a major process that's used in order to make sense of the evolution of bacteria. As well, we can look for genetic homologies. So when we talk about a homology, homo means same, so it's the study of the sameness, if that helps. We say that DNA is homologous if we see identical sequences of nucleotides in the chromosome. So if there's an exact match, then this piece of DNA and that piece of DNA are considered homologous. Sometimes there are missing gaps. Oops. Sorry, don't know what happened there. Sometimes there are missing gaps where, not missing, but where this gap is different. So these show greater homology than this one does, because this place here, this stretch of DNA, is not identical to this one. And so by looking for these stretches of DNA that are identical, then we can make more and more statements about how closely related the two individuals are. So here's a third idea where this is a different piece and this is a different piece from this one. So we would suggest that this third piece, that while this is this first one, they are the same species, then the more non-homologous or non-same stretches of DNA that we find, then the greater the likelihood that they are less closely related. So the more homologous they are, the more closely related they are. The less homologous they are, then the less closely related they are. But this is really expensive to do all of this, to be able to get all of the DNA collected for every species that we're trying to manage. So instead, the work of genetic, of people who are doing systematics through genetic homology, their work is to develop what are thought of as proxy measures. So some easier to gather data that will tell us maybe not the whole story, but get us a little bit closer to the truth so that we can afford or have time to do all of this systematics work. And so in the next slideshow, we will take a look at some of those techniques of genetic homology. Okay, so that's it for this one. You know what to do if you have questions. Come on.